So it's my pleasure today to introduce Janet Yeo, who'll be giving her completion seminar. Some of you may know Janet is originally from Malaysia, but did her undergraduate at Melbourne Uni. She started her research career here at WEHI, completing her honours year with Gabrielle Beltz, working on lymphoid tissue inducer cells, if I remember correctly. After that, she wanted a change in research directions and decided to start a PhD with me, working in RNA biology, with Gabrielle continuing on as a co-supervisor. So Janet had great ambitions at the start, choosing a very ambitious project. She really seized upon this RNAi screen that we're thinking about. Unfortunately, it was a little bit too ambitious for a PhD project. But fortunately for Janet, she had the foresight to think of a good backup project quite early on. And that's just what she's going to be telling us about today. So this backup project was about, she decided to characterize a novel RNase 3 protein that we had just identified in the lab, which we didn't know anything about. Despite a few hiccups, she's really driven this project with great gusto, and along the way has established a number of really fruitful collaborations with labs around Melbourne. This project has turned out to be quite interesting. It turned out to involve the mitochondrion, and that's what she's going to be telling us about today. Janet.
Thank you for the very kind introduction, and if I can get this working. So today I'll be talking about part of my PhD project, which was to investigate this protein that Mark has mentioned, Mrpl44, and its role in mitochondrial genome regulation. So first of all, a tiny bit of background on the mitochondria. The mitochondria is a very unique organelle within the cell. It is thought to have been evolved from a proteobacteria that is engulfed by another cell, forming what are now known as eukaryotic cells. So the proteobacteria is thought to be able to generate ATP, and I'll talk about that in more detail later in the presentation. But the mitochondria itself is a double membrane organelle. It has an outer membrane and an inner membrane. The outer membrane and the inner membrane of the mitochondria encloses the intermembrane space, and a lot of different proteins and factors are localized to this intermembrane space. The inner membrane of the mitochondria encloses the matrix of the mitochondria, and is also where the electron transport chain resides. The matrix of the mitochondria houses the mammalian mitochondrial DNA. So the mitochondria is quite unique among organelles within the cell, because apart from the chloroplast, it is the only other organelle that has its own set of genome that is completely separate from the nuclear genome. And this mitochondrial DNA actually bears more resemblance to bacterial genome compared to its nuclear counterpart. The mitochondria is involved in a variety of different cellular processes, including cell signaling, cell differentiation, cell death, the control of cell cycle, and cell growth. But perhaps the most well-known function for the mitochondria is its role as the cellular power plants. So as I've mentioned before, the inner membrane of the mitochondria houses the electron transport chain. The electron transport chain consists of 5 complexes that performs oxidative phosphorylation to generate ATP, which is used as an energy source for the cell. Going back to the mammalian mitochondrial DNA, as I mentioned before, it bears a closer resemblance to bacterial DNA. It is a circular, double-stranded DNA structure, and it encodes for 13 different messenger RNAs, 22 transfer RNAs in green, and 2 ribosomal RNAs in blue. The messenger RNAs that are encoded by the mitochondrial DNA encodes for crucial subunits of the electron transport chain. Going back to the electron transport chain, it is situated in the inner membrane of the mitochondria, and it is made up of 5 different complexes: complex 1, complex 2, complex 3, complex 4, and the ATP synthase. It is made up of around 90 proteins, and the majority of these proteins are actually encoded by the nuclear gene. However, 13 of the subunits are encoded by the mitochondrial DNA. So ND1 to 6 encodes for parts of complex 1, cytochrome B encodes for parts of complex 3, CO1 to 3 encodes for parts of complex 4, and ATP 6 and 8 encodes for parts of the ATP synthase. Now, the subunits that are encoded by the mitochondrial DNA has to be translated within the mitochondria itself, and this occurs within the matrix of the mitochondria. So the mitochondria actually has its own translational machinery, and the mammalian, well, the mitochondrial DNA itself actually encodes for the RNA components of the mitochondrial translation machinery. So the tRNA and the ribosomal RNAs forms the RNA components, whereas the protein components of the mitochondrial translational machinery is encoded by the nuclear genome. The mitochondrial ribosome, again, is an entirely separate species compared to its cytoplasmic counterpart. The mitochondrial ribosome is made up of a 39S large subunit and a 28S small subunit. The large subunit of the mitochondrial ribosome is made up of the mitochondrial DNA-encoded 16S ribosomal RNA and around 50 nuclear-encoded mitochondrial ribosomal proteins, annotated as Mrpl. The small subunit is made up of the 12S ribosomal RNA, again, mitochondrial DNA encoded, and around 30 nuclear-encoded mitochondrial ribosomal proteins, annotated as Mrps. Now, the proteins that are associated with the mitochondrial ribosome has been quite extensively studied, and perhaps the most extensive study on the proteins that are associated with the large subunit of the mitochondrial ribosome was done by this group in 2001, where they pulled down the large subunit of bovine liver mitochondria and annotated or sequenced the proteins that are associated with it. Some of these proteins have previously been annotated, some of these are novel, and they came up with a list of around 50 proteins that are associated with the large subunit of the mitochondria. So this is just a small part of what they did. So they did a sequence comparison between the proteins that are associated with bovine liver mitochondria to the sequences in humans, flies, worms, yeasts, bacteria, and plants. And what they found was that there's quite a lot of conservation in these proteins across different species. But I'm not going to talk about all 50 proteins that have been identified today. Today we're just going to focus on Mrpl44. So why Mrpl44 out of this list of 50 proteins? We first came across Mrpl44 when we were looking for proteins that have a sequence homology or a protein domain that is similar to the RNC domain in Drosha. So an RNC domain is basically RNase 3 in red and the double-stranded RNA binding motif in green. Drosha has been found to be crucial in the regulation of small RNAs, along with another protein, dicer, which also contains this RNC domain. They're known to regulate the biogenesis of small RNAs, in particular microRNAs. And in more recent years, well, in the past decade, these two proteins have been utilized in RNAi technology in the generation of silencing RNA. Drosha and dicer are crucial for the development of cells. So if you knock out either Drosha or dicer in mice, it is embryonic lethal. Drosha and dicer are known as RNase 3 family proteins, so proteins that have this RNC domain, and are thought to have been evolved from RNC in E. coli. So RNC is a much more simple protein compared to Drosha and dicer, only containing this RNase 3 domain and double-stranded RNA binding motif. And it usually works as a dimer to recognize double-stranded RNA and cleave it to generate products that have a 5' phosphate and a 3' hydroxy termini. So when we align the RNC domains of RNC in bacteria to Mrpl44 in mice and Drosha in mice, we found that there's quite a lot of conservation between the three different proteins. So highlighted in bold red are the residues that are conserved across all three of the proteins, whereas in just normal red are the residues that are conserved between Mrpl44 and Drosha. And it is quite remarkable, considering the fact that RNC is a bacterial protein and Mrpl44 and Drosha are mammalian proteins. So given the fact that Mrpl44 has this characteristic RNase 3 domain, a double-stranded RNA binding motif, and the fact that it is, as of now, the only other known protein in mammals that have this RNC domain, we thought perhaps it plays a role in the processing of RNA. And this leads me to the hypothesis of my PhD, which is that Mrpl44 plays a role in the processing of RNA. The aims of my project: 1, to determine the expression and localization of Mrpl44. 2, to determine the potential models for Mrpl44 function. 3, to determine whether Mrpl44 plays a role in the regulation of the mitochondrial genome. And 4, to determine the effect of Mrpl44 on cellular respiration. Going into my first aim. So despite the fact that Mrpl44 was pulled down in this complex that associates with the large subunit of the mitochondrial ribosome, it has also been pulled down using various nuclear and cytoplasmic baits, suggesting that it might localize perhaps not exclusively to the mitochondria, but also to the nuclear compartment or the cytoplasmic compartment of the cell. So to determine where Mrpl44 is localized to in my cell type, I transduced my cells with an Mrpl44 GFP fusion construct. So the Mrpl44 fused to the GFP allows us to very easily localize the expression of the protein, because whenever Mrpl44 is expressed, GFP is expressed along with it. So the localization experiment is done via immunofluorescence microscopy. Prior to imaging, I stained the cells with Mitochondrial red for the mitochondria and DAPI for the nucleus. And this first slide is the results from NIH3T3 fibroblasts. So in the red, the Mitochondrial red stains for the mitochondria of the cell. The blue, DAPI stains for the nucleus. The green is where my Mrpl44 GFP is being expressed to. And when I overlaid the 3 channels, what I saw was that Mrpl44 GFP is expressed almost exclusively in the mitochondria of the cells. I then went ahead to look at the expression of my Mrpl44 GFP in 2 different cell lines. One, the DP24 mammary epithelial cell lines, and 2, the N2A neuronal fibroblasts. And again, the colors are the same. The red stains for the mitochondria, the blue for the nucleus, the green for Mrpl44 GFP. And again, what I saw was that Mrpl44 GFP is localized almost exclusively to the mitochondria of the cells. So we concluded from here that Mrpl44 GFP is expressed exclusively in the mitochondria, which contradicts with it being able to be put down with nuclear and cytoplasmic baits. However, this is only just an overexpression construct, so I decided to look at whether the endogenous Mrpl44 is localized to the mitochondria as well. To do this, I used wild-type 3T3 cells, and I subfractionated it into a couple of different fractions. So the first fraction, the whole cell extract. Second fraction, the cytosol and heavy membrane fraction without the nucleus. The third fraction, just the nuclear fraction. The cytosol without the heavy membrane or the nucleus. And I further subfractionated the mitochondria into 2 different fractions. The first fraction, containing the outer membrane of the mitochondria and the intermembrane space. And the second fraction, containing the mitochondrial mitoplast, which is the inner membrane of the mitochondria and the matrix of the mitochondria. I then did immunoblotting on the different fractions, and using GRP75, which is a nuclear-encoded mitochondrial matrix protein as my localization control, I blotted the fractions and saw that GRP75 is expressed where you would expect the mitochondria to be at. So in the whole cell extract, in the cytosol and heavy membrane fraction, not in the nucleus, not in the cytosol, a bit in the outer membrane and inner membrane fraction of the mitochondria, but predominantly in the mitoplast of the mitochondria. I then blotted the different fractions with an antibody against endogenous Mrpl44 and found a similar distribution to GRP75. So Mrpl44 is expressed in the whole cell extract, in the cytosol and heavy membrane fraction, not in the nucleus, not in the cytosol, a tiny bit in the outer membrane and inner membrane fraction, but predominantly in the mitoplast of the mitochondria. So from here, we concluded that the endogenous Mrpl44 is localized predominantly within the matrix of the mitochondria. To summarize the first part of my talk, what I found was that Mrpl44 is expressed predominantly in the mitochondria, and more specifically, it is localized to the matrix compartment of the mitochondria. Moving on to the second part of my talk, to determine potential models for Mrpl44 function. Now that we've ascertained the fact that Mrpl44 is localized to the matrix of the mitochondria, and this is coincidentally where the mitochondrial DNA is situated at, we thought perhaps it plays a role in the regulation of the mitochondrial DNA. Given the structure of Mrpl44 with this RNase 3 domain and double-stranded RNA binding motif, we hypothesized that perhaps Mrpl44 works as a traditional RNase 3 enzyme that is capable of recognizing and cleaving double-stranded RNA, or perhaps it works as a double-stranded RNA binding protein, which either stabilizes the RNA or allows another RNA to come along to cleave it. But before I go into the potential ways in which Mrpl44 might work, a little bit of background on transcriptional processing in the mitochondria. The mitochondrial DNA, one of the unique features that it has is that it doesn't have any introns. So when the genes are being transcribed, they're actually all being transcribed as one long polycistronic transcript. So initiation of transcription is mediated by pol RMT, along with TFEM and TFB1 and 2. Elongation is mediated by PTCD2, and all the genes that are encoded by the mitochondrial DNA are transcribed as this long polycistronic transcript. So the ribosomal RNA, the transfer RNAs, the messenger RNAs are all just transcribed in the same manner. However, the relative abundance of these different RNA species are actually significantly different, and there are a couple of ways in which this can be mediated. So the processing of the different RNAs could be different. The maturation rate could be different. Degradation of the different RNA species could be different. Today, I'm just going to be talking about the post-transcriptional processing of the RNA. And the predominant model for post-transcriptional processing within the mitochondria is known as the tRNA punctuation model. So most of the RNAs within the mitochondrial DNA, the messenger RNAs and the ribosomal RNAs, are interspersed with transfer RNA genes. So as the genes are being transcribed into RNAs, most of these messenger RNAs and ribosomal RNAs are flanked by a tRNA. The tRNA are recognized at the 5' end by this RNase Z complex made up of 3 different proteins, and at the 3' end by this RNase P complex made up of 2 different proteins. The RNase P and the RNase Z work to cleave the 2 parts of the tRNAs, thus releasing the tRNA from the long polycistronic transcript. Now, when the tRNAs are being released, the flanking messenger RNAs and ribosomal RNAs are being released as well. And this allows them to be processed differently. So ribosomal RNAs would undergo nucleotide modification, tRNAs undergo CCA addition, and messenger RNAs will undergo polyadenylation. Of course, as with most biological models, there are exceptions to the rule. So these messenger RNAs can actually be cleaved in the absence of RNase P and RNase Z. Now, in some cases, it's quite understandable why it's not dependent on RNase Z or RNase P. For example, CO3 and ATP6 do not actually have a tRNA in between them, suggesting that there's some other form of transcriptional processing or post-transcriptional processing that's occurring. However, as of now, no candidate has been identified, and there are a couple of roadblocks towards the identification of mitochondrial RNases. One, you do not really need a lot of mitochondrial or a lot of RNases to be biologically active. 2, RNases are usually pretty small. And 3, the localization of RNases, particularly in the mitochondria, is not exclusive. So if you can find the RNAs in the mitochondria, most of the time you can actually still find them in the cytoplasm. So these are the roadblocks. But given the fact that Mrpl44, as I've shown, is expressed exclusively in the matrix of the mitochondria, it makes it quite a prime candidate for the regulation, not only for these messenger RNAs that are independent of RNase Z and RNase P, but also the regulation of the mitochondrial genome as a whole. So potential models for Mrpl44 function. Given the simple structure of Mrpl44 and its similarity or resemblance to bacterial RNase 3s, it is quite possible that it functions as a dimer. And this dimer is capable of 1, recognizing and cleaving secondary RNA structures, such as the tRNAs, and 2, recognizing and binding to RNA, allowing the other RNases to come along to cleave it. It is also quite possible that Mrpl44 forms a complex. And this complex, perhaps the prime candidate for which it complexes with, is a large subunit of the mitochondrial ribosome. But it is also possible for Mrpl44 to form complexes with other RNA processing proteins, for example, RNA helicases or PAX domain proteins, to form complexes that are more similar to either Drosha or Dicer. Today, I'm mainly going to be focusing on my research into whether Mrpl44 forms a dimer and whether it binds to RNA in the mitochondria, and briefly touch on what it might complex with. So, does Mrpl44 form dimers? To answer these questions, I transfected 293 T cells with 2 combinations of overexpression constructs. 1, Mrpl44 GFP and Mrpl44 flag, and 2, Mrpl44 GFP and flag back. Flag back is a mitochondrial outer membrane protein, and I utilize this as my negative control. The idea here is, so the green is my Mrpl44 GFP, the red my Mrpl44 flag. If Mrpl44 is to form a dimer, if you were to pull down the whole cell lysate from this transfection with a flag antibody, you should theoretically be able to pull down Mrpl44 GFP along with it. So if you immunoblot the IP fraction, you should be able to see a band for GFP. Whereas if you pull down with flag back, which has no known interaction with Mrpl44 GFP, you shouldn't be able to pull down Mrpl44 GFP along with it. So there'll be no GFP band. So this is my flag IP with anti-flag blot. 1 for Mrpl44 GFP and Mrpl44 flag. So Mrpl44 flag is being pulled down quite nicely. 2, Mrpl44 GFP flag back. Flag back is pulled down quite nicely. When I blotted this same lanes with anti-GFP antibody, first lane, Mrpl44 GFP, Mrpl44 flag, Mrpl44 GFP is being pulled down. Second lane, Mrpl44 GFP flag back. Mrpl44 GFP is not pulled down. And most of the Mrpl44 GFP is in the unbound fraction of the IP. So from here, we concluded that Mrpl44 does form dimers. Next, I looked into whether it binds to RNA. And to do this, I utilized 3T3 fibroblasts that I transduced with Mrpl44 flag and flag back, again, as my negative control. So I obtained the 3T3 cells, obtained the mitoplast fraction, IP'd the mitoplast fraction with flag agarose beads, extracted the bound RNA from the beads, and did a reverse transcription, and did a qPCR on mitochondrial genes. So again, the idea here is, if my Mrpl44 dimers does bind to RNA, when you pull down my Mrpl44 flag with a flag antibody, you should be able to bind, or you should be able to pull down the RNA that associates with my Mrpl44 dimers. So you should be able to see an enrichment with my Mrpl44 flag IP. So this is a qPCR of my IPs. So in the gray is my flag IP, and in the red is my Mrpl44 flag IP. And as you can see here, in the mitochondrial genes that are investigated, there is a strong enrichment with the Mrpl44 flag IP compared to my flag back IP, with significant enrichments in the 12S, 16S, CO1 mitochondrial genes. Perhaps most significantly, there's this huge enrichment in the 16S ribosomal RNA, and this correlates with the literature that states that Mrpl44 associates with a large subunit of the mitochondrial ribosome. I then went ahead to do an endogenous Mrpl44 IP to see if I can see a similar sort of enrichment. And to do this, I IP'd whole cell extracts from wild-type 3T3 cells with an Mrpl44 antibody and an Mrpl11 antibody. So Mrpl11 is another known protein that associates with large subunit of the mitochondrial ribosome, and it has no known RNA binding function, so that works as my negative control. So again, as you can see here in the gray is my Mrpl11 IP, in the red, my Mrpl44 IP. There's again an enrichment in the RNAs of the mitochondrial genes that I looked at, with significant enrichment in the 16S, CO1, ND4, and cytochrome B. And again, there's a statistically significant enrichment of the 16S ribosomal RNA. So to summarize the second part of my talk, what I've shown you is that Mrpl44 forms dimers. It associates with a large subunit of the mitochondrial ribosome, and this is from the strong association of Mrpl44 with the 16S ribosomal RNA, and also that it binds to mitochondrial DNA-encoded RNA. The third aim was to determine whether Mrpl44 actually plays a role in the regulation of the mitochondrial genome. So to investigate this, I performed an Mrpl44 knockdown. And to do this, I generated shRNAs against Mrpl44 using a non-targeting control as my controls. With the shRNAs that I've generated against Mrpl44, we observed around an 80 to 90% knockdown via qPCR. So in the gray is my control shRNA, in the blue is my Mrpl44 shRNA. And these are done in, again, 3T3 fibroblast cells. And at the protein expression level, Mrpl44 is not completely knocked down, but there's a sizable reduction. GRP75, again, is used as my loading control. So this is a qPCR between my control shRNA 3T3 cells and my Mrpl44 shRNA 3T3 cells. In the gray is my control, in the blue is my Mrpl44 knockdown. And these are the genes that I looked at. And what you can see is that when you knock down Mrpl44, the RNA expression of the mitochondrial encoded genes are decreased. Some of them statistically so, but there's a trend where it's decreased, even if it's not statistically significant. When I looked at the protein expression level, again using GRP75 as my loading control, most of the mitochondrial encoded proteins are also decreased when you knock down Mrpl44. So this shows that Mrpl44 does play some form of role in the regulation of the mitochondrial genome. So from my previous slide, I've shown you that Mrpl44 knockdown decreases both the RNA expression level and the protein expression level of mitochondrial encoded genes. So a decrease in RNA expression level quite possibly translate to a decrease in protein expression level. But we also wonder whether this decrease in protein expression is due to the fact that when you knock down Mrpl44, protein translation, the machinery itself is also being affected. So to answer this question, I performed a mitochondrial translation assay with Luc Formosa at La Trobe University, where I radio-labeled my mitochondrial translation products with 35S-methionine, and I did so for 120 minutes. And to stop cytosolic protein synthesis, I introduced amytine to the cell cultures. So theoretically, all the radio-labeled protein products that we would see in our gel would be products that are from the mitochondria. And this is the kind of thing that you would see. So we obtain whole cell extracts from 0 minutes, 30 minutes, 60 minutes, and 120 minutes. This is my control shRNA sample. This Kumasi stain works as my loading control. So at 0 minutes, you wouldn't expect to see anything because no mitochondrial translation products are being made. But you would expect to see the intensity of the bands to increase over time, and also the number of bands to increase over time as more and more proteins are being made, which is what you see here. With my Mrpl44 knockdown, what you can see is that, in general, the intensity of the bands are decreased, and also the number of bands are decreased as well. To do further analysis on this, I normalized the intensity of the bands in both of these plots to the 30-minute COX-2 band in my control knockdown. And this is the kind of results that we got. So ND1 to ND6 encodes for parts of complex 1, cytochrome B, parts of complex 3, CO1 to 3, parts of complex 4, and ATP6 and ATP8 for the ATP synthase. And what you can see here is that when you knock down Mrpl44, these subunits are not being made at all, whereas the subunits that are being made are generally decreased. The expression levels are generally decreased compared to the control. And even with the subunits that are being made, the rate of assembly, so ND1, ND2, cytochrome B, CO1, CO2, ATP6, and ATP8. And the time points are 30 minutes, 60 minutes, and 120 minutes. They're being made at a later time point compared to the control shRNAs. So it's not just that the protein expression levels are decreased, but the rate of assembly of the subunits that are being made are decreased as well. So to summarize this part of my talk, Mrpl44 knockdown decreases RNA expression levels of mitochondrial encoded genes. It decreases protein expression levels and also the rate of assembly of mitochondrial encoded proteins. The last aim of my project, or the last part of my talk, was to determine the effect of Mrpl44 on cellular respiration. Going back to the mammalian mitochondrial DNA, the messenger RNA encodes for crucial subunits of the electron transport chain. So these are the subunits that are encoded by the mitochondrial DNA. So apart from complex 2, almost all of these complexes that form the electron transport chain do have mitochondrial encoded proteins. As I've shown you in the previous part of my talk, when you knock down Mrpl44, you disrupt the translation of these subunits. So theoretically, or you would presume that there would be some form of effect on cellular respiration. A brief refresher on the electron transport chain and oxidative phosphorylation. So the electron transport chain resides in the inner membrane of the mitochondria. It is made up of these 5 complexes. And as electrons are being transferred from one complex to another, protons are being pumped out into the intermembrane space of the mitochondria. As the protons are being pumped out, it forms a proton gradient, which allows the protons to be pumped back via the ATP synthase, which allows the ATP synthase to convert ADP into ATP. And ATP is used as the energy source of the cell. To measure whether Mrpl44 knockdown affects the metabolic rate of the cells, I performed a seahorse analysis with Matthew Burt in MCRI. And the way that we measured the respiratory capacity of the cells is to measure it during resting phase, and also when you add different inhibitors to the cells. The first inhibitor that we add is oligomycin, which is an inhibitor of the ATP synthase. And this allows us to differentiate between electron transport chain linked protons, and allows us to differentiate between electron transport chain linked respiration and proton leak. The second inhibitor that we added was FCCP, which depolarizes the inner membrane of the mitochondria, and it allows you to theoretically raise the respiratory capacity of the cell to its maximum rate. The last combination of inhibitor that we added was rotenone and antimycin A. So rotenone is an inhibitor of complex 1, antimycin A an inhibitor of complex 4, and this allows us to differentiate between respiration that is mitochondrial linked and non-mitochondrial linked. And this is the kind of graph that you would generate from that particular kind of analysis. So at resting phase, this is my control knockdown. At resting phase, oxygen consumption rate, OCR, relative to cell number is around 75%. When you add oligomycin, so you inhibit ATP synthase, OCR decreases. When you add FCCP, you depolarize the inner membrane of the mitochondria, thus raising the OCR to its theoretical maximum. And then when you add antimycin A and rotenone, which inhibits complex 1 and complex 4, you again bring it down. So that's non-mitochondrial linked oxygen consumption rate. When you knock down Mrpl44, what you see is that across the board, the oxygen consumption rate is severely and significantly decreased. A resting phase is decreased. When you add oligomycin, it drops. When you add FCCP, you can't even increase the OCR by much. And then when you add antimycin A and rotenone, it again decreases. From this kind of graph, you can come up with these kind of analysis. So you can look at the maximum rate of respiration, the spare respiratory capacity, and the extracellular respiratory capacity of the cells. Again, in the gray is my control knockdown. In the blue is my Mrpl44 knockdown. So maximum respiration rate is significantly decreased when you knock down Mrpl44. Spare respiratory capacity is significantly decreased as well with Mrpl44 knockdown. Extracellular respiratory rate is not affected, thankfully, and this is expected due to the fact that it's not linked to the mitochondria at all, and you do not expect Mrpl44 to affect non-mitochondrial linked respiration. So to conclude this last part of my talk, what I found was that Mrpl44 knockdown decreases the mitochondrial-linked oxygen consumption rate of the cells. It decreases the maximum respiratory rate and the spare respiratory rate. It does not affect non-mitochondrial-linked respiration. To summarize my talk, the conclusions that I drew from my PhD project is that Mrpl44 is localized to the matrix of the mitochondria. It forms a dimer and is very likely to associate to the large subunit of the mitochondrial ribosome. And it is likely to bind to RNA in the mitochondria. What I found was that Mrpl44 affects the mitochondrial RNA and protein expression level, as well as the rate of assembly of mitochondrial proteins and the overall respiratory capacity of the cells. So what has this got to do with anything? More and more recently, it has been shown that defects in mitochondrial translation causes a wide range of diseases from cardiomyopathy to infertility. When the defect is found in genes that are encoded by the mitochondria, it's pretty straightforward to identify them due to the fact that the mitochondrial DNA is so small. However, more and more recently, nuclear-encoded mitochondrial proteins, particularly proteins that are involved in mitochondrial translation, has been found to cause a wide range of diseases. In particular, Mrpl44 was found in January 2013 to cause mitochondrial infantile cardiomyopathy. And this disease is caused by a mutation in the double-stranded RNA binding motif of Mrpl44. The mutation causes a severe complex 4 deficiency that can be rescued when you introduce wild-type Mrpl44. Now, this disease is caused by just one mutation in just one domain of Mrpl44. Presumably, mutations in different parts of the protein would also cause different kinds of diseases. So it's quite crucial that we understand how Mrpl44 actually works so that we're better prepared when these mutations happen. And this leads me to my acknowledgments. I'd like to thank my supervisor, Mark and Gabriel, who allowed me the freedom to pursue this project. My lab members for various help, either in the lab or in terms of moral support. My committee members, Steve, my committee chair, Ross, and Ruth. Ruth in particular has been my gateway to the world of mitochondria. She introduced me to different people who worked in the mitochondrial field, including David Thorburn, whose student Matthew Bird I worked with to perform the seahorse analysis, Mike Ryan in Latrobe, whose student I worked with to perform the mitochondrial translation assay, the people in molecular medicine. In the last part of my PhD, I performed a lot of my protein work with the Cancer and Hematology Division. In particular, Django has been very kind in allowing me to perform the majority of my protein-based experiments with him. Kaylin, who has contributed to quite a lot of discussions about proteins, Dina, Mark, and people in the imaging facility, who perhaps helped me produce the prettiest pictures of my PhD, people in radiation and instruments, people in the fax lab and animal techs, and of course my family and friends, in particular my father, who actually flew all the way from Malaysia to come and attend my talk. So, and thank you all for your attention.
There's plenty of time. To stop that.
So when you knock down Mrpl44, do you see any effects on general cell functions such as survival or proliferation?
I didn't actually measure any of those, but just by eye, with the control knockdown and the Mrpl44 knockdown, they seem to grow at the same rate. Yes.
I think that in one of your early slides you showed a comparison of the L44 to some eukaryotic ribosomal proteins. Is that right? About slide number 4 or 5 or something?
So Mrpl44 is found in quite a few different eukaryotic cells.
No, but there were some homologs.
Yes.
That might have been in the eukaryotic
Yes.
ribosome.
Yes.
Can you go back to that one?
Sure. This one?
Yeah, that's right. So, you know, the structure of the mammalian large and small subunits of ribosome, I think, has been solved. And I was just wondering, where does the homologous protein reside in the large subunit of the mammalian ribosome?
As in where
I was just wondering if you
specifically?
get clues.
Right.
You may not have looked, but
Right.
I was just wondering whether the ribosomal mitochondrial protein would lie in an analogous position to the cytoplasmic, ribosomal
Right.
equivalent.
Right.
Like if it lay and was
Yep.
in its condition, it was a dimer, that would support your
Yes.
findings.
I'm not aware that there is a homolog in the cytoplasmic ribosome. So these are all mitochondrial ribosomal proteins. But in terms of where Mrpl44 is actually situated within the large subunit of the mitochondrial ribosome, it has been posited that it's pretty much buried within the structure itself. So it's not on the surface.
Right. Oh, okay. Right. So there is. Yeah. Right. Good.
Yeah.
If that were the case, how would you be able to IP it then?
Perhaps when you do a whole cell lysis, you denature part of the complex to allow the protein to come out. I don't know. Maybe.
I just want to add another question about the dimer. When you do the IP, if you're pulling down larger complexes that might be held together by RNA, then you would expect to see the GFP tagged one while pulling down the flag tagged one. So how do you know you're not pulling down a larger complex that's a lot more complicated than just a dimer?
Yes. So it's probably unlikely that Mrpl44 just forms a homodimer. It is very likely that it forms like a large complex, and the IP that we actually are pulling down is actually pulling down like a large complex. We just haven't gotten around to further teasing out what that interaction is. It is very likely that you're right. So Mrpl44 could be in one part of the complex, and another Mrpl44 in another part of the complex, and you're just pulling down the whole thing together with it. However,
Can you comment on the column work?
the HPLC work. So I did a HPLC subfractionation assay, and what we found was that Mrpl44 is not just doubling in size, it actually is distributed in larger sizes than just twice of Mrpl44. And this, again, suggests that it exists in a larger complex rather than just a homodimer.
Yeah, I suppose my question was about the dimer as well.
Yeah.
And you didn't really, I think, show data to say that it was specifically a dimer. I was wondering if you could do, say, for example, blue native page to get higher order structure information.
Sure.
And
So what?
I was also wondering, had you tried co-IP of Mrpl44 to see also what comes down with it?
Co-IP of Mrpl44 with
So pulling it down and then looking for interacting partners
Yes.
in an endogenous setting?
not in an endogenous setting. I did an IP of flag tagged Mrpl44, and I did a couple of runs of mass spec with it. But the results are pretty dirty. We did pull down a couple of RNA processing enzymes along with it, which I didn't show in this, which suggests that it might form a complex that is actually more to its RNA processing than just purely associating with a large subunit of the mitochondrial ribosome.
So the fusion proteins, or the GFP fusion protein, or the flag tag protein that you used for your to show that they form dimers, did you prove that they are functional? So when you express them in the Mrpl44 knockdown cells, do they restore normal mitochondrial activity?
I have not done those yet, but with the Mrpl44 GFP, I did overexpression. I did a similar analysis to the knockdown experiments and saw that when you overexpress Mrpl44, RNA expression level is increased, protein expression level is increased, which indicated that it's functional.
And mitochondrial activity is increased as well?
The cellular respiratory capacity is increased when we did a seahorse analysis, but that results, Matt and I, it requires further investigation. Yes.
Is dimerization RNA dependent, or could you digest the RNA and see whether you still get dimers?
Is the dimerization RNA dependent? Good question.
RNA in the sample to
Yes.
sequence.
Good question. I haven't actually done that experiment yet, but it is a pretty good experiment to try to do. Yes.
I've got two questions. The first is, your hypothesis is the dimer is leading to cleavage of this polycistronic mRNA. Have you looked at that biochemically? I don't know the practicalities of it, but running a gel with and without?
So we tried cloning the junctions that are flanking the tRNA punctuation exception models, and we tried doing it in an in vitro processing assay. But because we are really just shooting in the dark with this, I didn't actually see any cleavage. that assay actually requires more optimization because we were trying to modify it from another assay that was in a different compartment of the cell.
And the second question is, if these proteins are involved in the mitochondria themselves, and you're measuring your respiratory rate per cell, wouldn't a fairer comparison be to measure respiratory rate per mitochondria if the per cell mitochondrial number had gone down?
Right. So this goes back to the question of whether if you knock down Mrpl44, whether mitochondrial numbers are being affected, yes? So we did a comparison between my Mrpl44 knockdown and my Mrpl44 overexpression. And we looked at the copy of nuclear genome to mitochondrial genome. So that's just a crude way of measuring. And we found that there was not any difference, statistical difference, when you actually overexpress Mrpl44 or when you knock it down compared to the control, suggesting that the number of mitochondria, or rather, the number of mitochondrial genome is actually not affected.
Congratulate Janet on excellent results.